Hello, yes. Are y'all ready for a quiz today? Yes. Y'all love quizzes. Said it, I couldn't hear you. So much noise in the room. A few, yes. All righty, here we go. We stopped off last time talking about the Golgi apparatus. So today we want to talk, I think I talked about lysosomes, Golgi, then lysosomes, and then this is just showing you there's a lysosome that is actually engulfed a worn out organelle. Over here, you can see food molecule coming in, lysosome fusing with the vacuole, and then digesting the food molecule into smaller usable units. All right, microbodies. These are similar to your lysosome. The biggest difference, because they basically function in the same way as far as performing hydrolysis type reactions, the biggest difference in a microbody is going to actually be in where the hydrolytic enzymes arise from, okay? So it's going to, the difference in the microbodies is going to be mainly in where the hydrolytic enzymes are going to come from. So we knew the hydrolytic enzymes in a lysosome, where do they come from? Okay, originally from endoplasmic reticulum, absolutely. Well, it says here, then the enzymes that are used in microbodies, they're basically coming from free ribosomes. So, in other words, these ribosomes were never associated with the endoplasmic reticulum. So the microbodies, or, yes, sir? That's what I was saying. That they come from free ribosomes that have never been associated with the endoplasmic reticulum. So these ribosomes are basically just kind of floating around in the cytoplasm. Okay, so they've never been associated. I didn't even know you were here. You out your spot. Confused me. Okay, so we said generated by free ribosomes. In lab, when you go to lab, not next week, I think it's the following week, you will be harvesting some microbodies, okay? The other word that you're going to know for microbody, or a type, a specific type, is called a peroxisome, okay? And there are peroxisomes that are found in the liver, okay? And their job is to hydrolyze hydrogen peroxide, okay? And it turns hydrogen peroxide into water and oxygen gas. Hydrogen peroxide is not anything that we want to accumulate in the tissues. It's going to be in the tissues, but we don't want it to accumulate in the tissues, and the reason being that we don't want hydrogen peroxide to accumulate in the tissues is because it causes necrosis, okay? When hydrogen peroxide, let me see if my pen will actually work today. It didn't even work the other day. Oh yeah, it's gonna work today. Yes, sweetie. Absolutely. Okay, so we said we don't want a huge amount of hydrogen peroxide to accumulate because it causes necrosis, okay? Yes, and that's the same, that kind of thing. All right, so the enzyme, specific enzyme's name is called catalase. So the specific enzyme that's going to hydrolyze the hydrogen peroxide, its name is called catalase. And so catalase will degrade or hydrolyze H2O2 Oh, I forgot I got to get rid of this. Mercy. It'll hydrolyze H2O2. Where's my little arrow? Uh-oh, go back. Oh, no, mercy. What the peanut butter and jelly? Oh, I guess we'll be moving on. But anyway. It's going to hydrolyze, H2O2 into water and oxygen gas, okay? Let me see, 2 times 2, there go 2, and then O2. That's 4, so maybe 2 of them. Did I get my calculations right? 1 oxygen, 2 of these? Okay, 2 times 2, 2 times 2 H's. We got 4 H's, right, there we go. And then 2 times 2 is 4 O's. 1, 2, 2. Oh, 4. There we go. I have to do my math, to make sure. So we got 4 hydrogens and 4 oxygens on both sides. There we go. All right. So we said, who's doing it? We said catalase. Yes, sir.
Spell catalase?
Yes, catalase. All right, and we said this is an enzyme that's going to be responsible for hydrolyzing or breaking down the hydrogen peroxide.
What was that process?
Means cell death, causing tissue death, yes. All righty, so then the vacuoles and the vesicles. Vacuoles and vesicles, we know that these are just simply membrane packages, okay? And usually the guy that's going to be responsible for unwrapping and also packaging things that are coming, so unwrapping things that are coming into the cell, packaging things, again, that are going outside the cell, that's going to be the Golgi body, okay? So anything coming into the cell, we know automatically is going to be unwrapped by the Golgi bodies. The main difference that we see in vacuoles and vesicles is the size and what they actually house, okay? So we're saying they're organelles too. The biggest thing, again, is size. Vacuoles are usually large membrane packages, and your vesicles, they are small membrane packages. So vacuoles, we said, are large membrane packages. Vesicles, we said, are small membrane packages. See, can I grab the pen?
If it comes into the cell and it's repackaged by the Golgi bodies, it's not just allowed to go free floating in the cytoplasm. Does it then repackage them for some reason?
I'm sorry, I'm writing, so I didn't hear a word that you said. I have a one-track mind. I'm old. All right, here we go. Now, what did you just ask me?
I said, the stuff that comes into the cell, when it comes in, it's unwrapped by the Golgi bodies.
Right. So, in other words,
Reroute?
depending on where it needs to go.
Okay.
Yeah.
It's leaving the Golgi body, it's going to have to be rerouted.
Right. Okay, so vacuoles, we know, generally contain solid particles, and our vesicles, they're going to contain your liquids, okay? So things that are in solution, so things that can be put into solution by water, for example. That's what's going to be inside of those guys. Okay, so we said there's some exceptions to these normal rules, though. Can somebody name me something that disobeys these rules? Okay, that vacuoles generally have solid things in and vesicles liquid. Yes?
In the plant cell, isn't the vacuole?
Correct. The large central vacuole of plant cells. We know that the large central vacuole of plant cells is liquid. Okay? So maybe it got rid of my pen. There we go. All right. So Inside the large central vacuole, we're going to have like water. In solution, we're going to have sugar, salt, and then also that's going to be present are your toxic substances that are used for protection. But the large central vacuole is an exception to that normal rule that we said that where vacuoles are going to contain things that were solid and vesicles would contain things that were liquid. And there's some other exceptions, but this is the only one that you need to know for the exam. Okay, let's talk about microtubules and microfilaments. Microtubules and microfilaments are protein threads that are found in the cell cytoplasm. And when you're reading your book, you'll probably read that there are actually three. I'm probably only going to talk about two. But in the book, you'll see actin filaments, you'll see microtubules, and you'll see intermediate filaments. So the microfilaments, the microtubules, and then an intermediate one. Microtubules, we know that these are the hollow threads. The word tubule leads us to believe that it's hollow. Okay, it's made of a protein that's called tubulin. And we'll take these microtubules and the intermediate filaments and the microfilaments, and all of those guys together will make up your cytoskeleton. So the microfilaments, again, are also called actin filaments, another protein thread. So we know that this one is much thinner than the tubule itself. It's not hollow. So again, we said that these two guys, along with the intermediate filaments, they help to make up the cytoskeleton. The cytoskeleton for the cell is similar to the way that your skeleton works for you. So that means that your skeleton does what for you? Structure. Good. Okay? And also protect certain things. That's what ours does. So it adds support and also movement. Absolutely. Say it again. Right. What about the red blood cell? I can't hear you. The cytoskeleton? Oh, you mean the skeleton, right. Well, this one doesn't do that, okay. I was doing a comparison, right. But, right, in certain areas of the skeleton, absolutely, where the marrow is in our skeleton, it's going to help create red blood cells, absolutely. Right, correct. Correct. And then here's my cute picture. And so here's the nucleus. What is this thing? Beautiful. What is that thing? What is that? What is that? ER, the endoplasmic reticulum. Yes, sir. They don't transport things, but they help with movement. And we're gonna talk about, we get now. It ain't holding it and transporting it somewhere. It's kind of basically giving it structure. But we're gonna talk about how movement takes place, okay? With the microtubules. There's a specific guy that we're gonna talk about. Okay, were y'all done with that? Anybody know what these guys are? Yes. This is just giving everything structure, but there are some microtubules that make up, for example, cilia and flagella, okay? So their job is for movement, okay? And we'll talk about how they work. Thank you very much. Appreciate it. All righty. So let's talk about cell movements. All right. Actin filaments and myosin filaments, they act in concert as far as like in skeletal muscle, for example, cardiac muscle, and it's when those two are pulled together by the myosin's arm. It says that's what causes a contraction to take place. But actin filaments, they help with the contraction of muscles, for example. Usually if you see actin, it's always going to be in this these helical chains. They give you a really pretty picture in your book because this is not going to be pretty at all. Okay? I am not an artiste. I did not get that bug. Okay? Kind of like that. Yeah. Anyway, you get the picture. I'm not an artiste. All right. So you normally have two of those chains that are twisted, and then you have, and we won't get into all of that, but actin actually has a site on it where myosin will actually bind, and myosin basically is another one that has two heads. One of the heads is where ATP binds, and then the other one will bind to the active site that's on myosin. And once you have this cross bridge form, then you have the ratcheting effect that shortens the muscle. But we won't, yeah. You want to talk about tropomyosin and all those, right. The tropomyosin is actually the thread that covers the active sites until it has been activated, where it rolls away, and then now myosin can bind. But anyway, we won't get into all that. Just know a little bit. All right. So muscle cells have many of these microfilaments, and they can contract very strongly. Microtubules, big guy, makes up the centrioles. Usually you have two pair of centrioles, and I'm going to give you a pretty picture of centrioles, but I'm going to give you my elementary picture of centrioles. Okay, so you have basically 9 triplets of microtubules. So there's these triplets of microtubules, and you'll see it in a minute. But anyway, they create this circular bundle. And notice that it's hollow in the center. So there's nothing running in the center of these guys. So this guy has what's called a 9 plus 0 arrangement of microtubules. Now, its job is to make spindle fibers, and we want to make spindle fibers. Why? Because we need to move chromosomes, okay? So these spindle fibers yes. This is called the microtubule organizing center for the cell. This and the asters is referred to as a microtubule organizing center for the cell. So they make spindle fibers, and these spindle fibers' job is to basically line up the chromosomes. So after well, the nucleus is dissolved at this point, but yes.
So is that why the book pictures that diagram pictures the centrioles as being out in the regular cytoplasm?
Right. They're always out. This is outside in the cytoplasm. That is correct. Right. Correct. And so these were, first, because you remember, we said the chromatin net is loosely coiled how long? 90% of the time. The other 10 is when it's in this chromosome. You got these guys, so when what happens is, if we were talking about mitosis, we knew that all this was in the nucleus, right? But then what happens in prophase, it says the nuclear membrane breaks down and disappears, and the chromatin net condenses into chromosomes. And the reason why we want to condense them into chromosomes is because, is it easy to move this without breaking off pieces? No. So it says we want to use those histone proteins that are associated with it, pack it up into these neat little nice packages so that we can evenly divide the DNA and make sure, each new cell that's being created is only going to get half of the DNA material and not, Right. Because we know occasionally it does happen where you get too many chromosomes, or too few chromosomes. Right. Okay. All right, so centrioles are composed of 9 triplets. So what we call this is a 9 plus 0. So there's 9 of these triplet things going around in a circular bundle, and it says 0 because there's nothing running in the center. It's hollow. And we always have a pair of these centrioles in the cell. We always have a pair. Let me get the picture up here. So here's a triplet. Okay, that's what I say. When you see my little picture, it'll make a little bit more sense. I just can't draw it like that. So I gave you my little elementary drawing. You mean to tell me this doesn't look like that? Who said no? That's all right. That's all right. I did the best I could. At least I tried. All right, so the word MTOC stands for what? Microtubule. Good. Right. The other name that you're going to see for this, there we go. Sorry. So the other name that you're going to see for microtubule organizing center, I guess they didn't do that part down there, huh? How about that? There we go. All right. So the other name that you're going to see for that is centrosome. You're going to see this word. And that may be an E in there. I can't remember. It's been a long time. Can't spell. Okay, so you're going to see that word centrosome. Know that means MTOC. And also know that this means what? We already got that up there. For animal cells.
So what's the centrosome?
What I just wrote up all up on the board. Okay. The centrosome is the microtubule organism center for the cell, made up of 2 parts. The centrioles and the acids. What does it do? Make spinofibers. Okay? So you're saying function? Is that what you're looking for? Okay? Okay, more movements. We have the microtubules also making up the cilia and the flagella. Okay, so we're saying on this one it says when these things get longer and shorter, that's how eventually we're going to line these guys up in the center. So, we just kind of keep moving these around until we get them all lined up evenly. So, because these guys are scattered, everywhere. So that was movement that way. Okay. With the cilia and flagella, it says now we have a circular bundle again, which we're going to talk about in a minute, but there's actually some more microtubules going to be running down the center. All right, so we said, because it says, look at the difference. It says cilia and flagella. Remember at the beginning when we first started talking about this, and we talked about the flagella of a bacteria. And the flagella of a bacteria, how did it move? 300 spins, right? And I said, well, when you think about cilia and flagella, what comes to your mind? And you said more of a whip-like motion, right? Well, here comes a whip-like motion because it says, and I'll put the picture up in a little bit, but when these doublets pass each other, okay? So in other words, we're going to have this guy running down the center of it. But it's like when those doublets pass each other, that's what's going to give the whip-like motion that you see in this guy versus that spinning motion. And we'll get to it in a minute, but anyway. All right, so it says, let's start with the cilia. Cilia are short hair-like structures. In lab the other day, they had you examine a tissue. Anybody remember the tissue? For 2 points on your last test. Can anybody tell me what the tissue was? Let me hear. You got to tell me the name. Y'all just guessing some boo-boo. Yes. I'll give you 1 point just because you know that on your last test. Pseudostratified, ciliated, columnar, epithelium. Okay? Pseudostratified, ciliated. Pseudo meant what? False appearance that there was more than what? One layer. Because the word stratified tells us what? More than one layer. Okay? Or layers. Okay? So if it's false appearance that there's more than one layer, it says that's the tissue that's lining the respiratory tract. Okay? Its job is to do what? Good. So all that stuff that we're breathing in, stuff that doesn't belong down in our lungs, it sweeps it back up. That tissue also have what are called goblet cells. And those cells make mucus, okay, in order to help trap that crap. And what do? You hawk up a loogie and you spit it on your neighbor's car. Not right, okay? So these guys kind of help sweep it all, back up so you can make a nice, good loogie, yes. They maybe not burn it, but what it does, it decreases mobility. Now, if you're smoking a cigarette and you're standing around oxygen, probably burn it all. I had a friend whose sister was on an oxygen tank, and everybody that came to see her, she begged them for a cigarette. And then finally she begged up on a lighter too. Now she in there in the bed on the oxygen. Thank God she didn't die. But anyway, so they hear an explosion in the other room, and they go in, and she's, black, everywhere. And she goes, I don't know what happened to this darn thing. She was alive, but she was like, I don't know what happened to this darn thing. It just blew up on me. So then, but under the bed, they find the cigarettes, the lighter, But yeah, she singed her eyebrows off. It singed her hairs, but it didn't travel down and burn, because she would have been good as, dead. But anyway. Yeah. All right, so here we go again. You said it was on the who? Unicellular paramecium that you drew off the back of the wall. in lab the other day. He had cilia, but he also had a what? He had a flagella too. Yeah. All right, so sperm, they don't go under cilia. I just kind of got carried away. Okay, they have a flagella, okay, and we know that these guys, why do they need a flagella? To get to the egg, which is a long way away, okay? Because of somebody, where do they have to go? Anybody know where the sperm have to go? He said to the egg, right? So where? Somebody tell me the pathway then.
Birth canal, the cervix, and into the fallopian.
Okay, so uterus, right? Fallopian tubes, good. Long way, long way. So all the way to the fallopian tube, then it travels down. Once you fertilize it, then it travels down and implants in the uterine wall, okay? So long way. All right, and then I go back to cilia again. Old people, that's me. All right, so cells that line the upper respiratory tract, they have cilia. Flagella, then I go back to flagella. I don't know what was going on. I just want you to know, late night, okay? It was. It's late night. I stay up late, you know. Stay up late. All right. It says both are composed of, where are your minds? I don't know what y'all do at night, but I was up studying. I'm in school, okay? So it says both are composed of what? 9 pairs. So what do I mean by 9 pairs? Similar to what I drew over here, right? These are the 9 triplets, okay? But instead of having triple, now we're going to have what? 2. So 9 pairs of microtubules, but it says then we've got one running down down the center, okay? And what happens is, again, when the doublets are passing each other, this is what's giving that whip-like motion that we see versus that spinning that we saw. Correct, a 9-2. So know the difference. Centrioles have a what? 9 plus what? 0, and then the cilia and the flagella have a 9 plus 2 arrangement of microtubules. So how many people have already started studying for the next test? Okay, good. Because you need to start studying now. Don't let it snowball on you. Don't let it snowball. All right, let me ask you a question. I'm even going to ask the question. How many people think, all right, how do we power life? You should already know. Good. All right, so we're going to look at a little brief thing about energy. All right, energy, that's the capacity to do work. And we want to look at the difference between potential energy versus kinetic energy. Potential energy versus kinetic energy. Can somebody tell me what's the difference between potential and kinetic? Okay, energy at rest versus energy at motion. Did anybody want to disagree with that? No, everybody's good. So which one is which? Don't answer it out loud. Which one is which? What's potential energy? Well, I got my hand on him. I want him to tell me what's potential. Yeah, what is it? So which one is it out of the ones that he gave me? At rest, I heard. Correct. And then kinetic energy, you said the energy what? In motion. Good. So we want to say kinetic energy it's moving, right? So that's going to be very important to you, especially when we start talking about what these mitochondria do. Okay? They basically touch on it because chloroplasts do too, and we'll mention it briefly, but we're going to get down and dirty with it when we start talking about, Yes, absolutely. And it happens in photosynthesis as well, but we're going to really talk about it in the mitochondria because of the fact that we're building so much energy in the mitochondria. All right, so it says energy is neither created or destroyed, it's just changed. That's the first law of thermodynamics. So energy is neither created or destroyed, it's just changed. This is October. Is today the 3rd or 4th? 4th? 3rd? Okay, and then it says, and even with that, we can't change. It says without some loss of usable energy, okay? So usually you get a little bit of heat or something, okay? So it says neither lost nor gained, then we can't change it without a loss. All right, October the 3rd. Say it again. Because you still use the heat. It's not lost. Is that what you mean? You show? Say it again then, in a different language. What you doing? Oh, that's so teeny. Always. Somewhere else. All right, here we go. ATP. Ask me after class. So maybe if you rephrase it. All right, ATP, we know it's adenosine triphosphate. Correct? Adenosine says that what is your base? Adenine, okay? How many phosphate groups do you have? What's the sugar? Good, ribose, I heard someone say from the very back of the room. All right, so we said that this is the energy currency for every reaction that's taking place that needs some energy. And we say the high energy bond is this last one, and they say it's a high energy bond because the last one can be easily cleaved off, okay? So it's harder to break it here, but it says this one can ease, so it's worth a little bit more energy. Not that we can't take it down to the monophosphate, but yeah. Okay, and then it says the phosphate group is easily removed. That's why it's high energy. And remember the phosphate, that's the PO4 that you guys drew all kinds of way on the test. For those of you who had a phospholipid. I was really surprised that some of y'all didn't stick with the Ho and the O. I got an O and an O. Okay, and then this last line just says that we can regenerate ATP from adenosine diphosphate and an inorganic phosphate. So it says we can take ADP plus P and generate ATP. So it says that the reaction and that's an arrow, a bad looking one, but it is an arrow. Okay, so it says we can go in both directions. Okay, ATP is often involved in what we call coupled reactions. So, and what we mean by that is the ATP is being broken down. It says the energy can be used to fuel some reaction that needs that energy. So it says it's often involved in what we call coupled reactions, where one reaction may be giving off some energy, the other one needs the energy in order for it to proceed, so it's going to be coupled with that type of reaction. So we have reactions that release energy, and we have reactions that actually need energy. Anybody know the names for those two types of reactions? Yes, sir. Okay. Right. So, and then the particular reactions as far as the energy would be endergonic and exergonic. So endergonic is going to need energy, exergonic is going to release the energy. So exergonic reactions, they release energy. So when we're breaking bonds, we know that anytime we break a bond, we're releasing the energy that was stored. So in other words, if we were looking at, like y'all said before, you talked about potential energy. We can also refer to it as stored energy, okay? So it says, for example, we could have A plus D yields C plus D plus some energy, okay? That would be an example of an exergonic reaction because we see the energy at the end. It's being liberated from this type of a reaction. And then an endergonic reaction, we said they require energy. One thing that we're going to look at is, as an example of exergonic, an exergonic reaction would be glycolysis, the whole thing, cell respiration. That's an exergonic reaction where we release and build tons and tons of ATP from breaking down one glucose molecule. Solar energy is involved in an endergonic reaction of photosynthesis, where we need the solar energy, the sunlight, in order to make glucose and oxygen, okay? So we need some energy in order to take the carbon dioxide and the water molecule in order to build glucose. So it says what are some of the functions? It says chemical work. We know that both of those things that we just talked about, that's chemical work. So synthesis, for example, making or building some bigger molecule, taking those smaller guys. This would be synthesis kind of like work. All right, transport work. If we're moving things across the membrane, especially against their membrane, against their natural tendency. So in other words, if we were looking at having ions on this side of the membrane and having ions on this side of the membrane, it says in order to, if we had something that looked like that, what do they want to do? Right, because it wants to bring the system to what word am I looking for? Equilibrium, absolutely, to balance, so equilibrium. So it says in order to keep those, or to, not that, to say that there aren't any potassiums or sodiums on this side, but in order to keep more sodium on one side and more potassium on one side, we're going to have to work against what their natural tendency to do, which is to bring the system to equilibrium. So we're going to have some energy, okay, in order to transport those ions where we want them to be. So we're looking at functions. And then it says mechanical work. You can't get your muscles to move without having some calcium involved, okay? So us making sure that we're pumping the calcium where we need it to be. So it does all these different things. So chemical work, transport work, mechanical work. And then also keeping those guys in a muscle, where they're supposed to be. So we got to transport some things. Okay, muscles actually moving. We got to have ATP bound here, and then it's hydrolyzed to ADP. When we release that phosphate group, that's what powers the head to attach to the myosin. So we got to have some ATP to, cause that head to cock. So it's called to cock the head. And once the head is cocked, then it can bind to the site, that's the myosin binding site, which is on the actin molecule. And then we eventually get that ratcheting effect, okay, when we actually release the ADP. All right, so enzymes. We also have to have some enzymes involved in all of these different reactions. We write it simply as, we show the beginning of the reaction. On the other side, we show the end of the reaction. But know that most reactions are grouped, okay? And it usually takes more than one enzyme in order to take us from what we see at the beginning of the reaction to what we see at the end, okay? So it says an enzyme, we only need a teeny-weeny amount, because the enzyme, does it ever get used up? No, they never ever get used up. So we only need a small amount of enzyme. Now, mind you, those enzymes can be denatured, which is the other thing that you're going to be doing in lab. So you're going to examine catalase. You're going to find out what denatures catalase. You're going to examine another enzyme that's called renin. Most enzymes usually have ASE at the end, but there's some of the older ones that were named prior to that, like renin, who doesn't have the ASE at the end of its name. But you'll find that it only takes a small amount to catalyze reaction after reaction after reaction. And it's because they never get changed. Okay. Okay, I told you I was a late night. Okay. Yes. Heat, pH changes, either being too acidic or too basic. All of those things can denature. Freezing, you can actually denature. Boiling can denature. Any extreme thing, yes. Because, the warmer, usually the better. But if we go beyond that operating range, the max temp, then boom, we're destroying it. All right, so it says enzymes, it says they bind tightly to their substrate. We know the substrates are generally what's on the front side of the reaction. So in other words, if we were looking at cell respiration, for example, we know the formula for glucose is C6H12O6 plus oxygen. It says the enzyme is going to bind to who? The substrate. Okay? Mainly this guy. Okay? This guy's going to be used, but this guy's the guy that we're talking about. So it says they're going to bind tightly to the substrate in the reaction. But the enzyme, he may be modified a little bit, but he's going to change the substrate into whatever the product is. What's the product that glucose is going to be converted to? Okay, ATP is the energy storage part, but what will he be converted to? Carbon dioxide. The oxygen will be converted basically into the water. Okay? And we'll talk more about that when we get there. Okay? Because we'll talk about the coenzymes and what they do too. Anybody, some people got that right on the test. Not very many. But got that, what a coenzyme does. Because they're often going to be involved in these reactions as well. So it says you have your reactants, A plus B, but then they're going to be changed to what? The products, C plus D. So the glucose is being converted to who? Carbon dioxide. So then we're talking about oxidation and reduction eventually. And it says that oxygen is being converted to who? Water. So we're saying glucose is being broken down or oxidized and it says that oxygen is picking up the hydrogen ions that the cab drivers were carrying around, and it's being reduced. Okay. It was a long night. All right. All right, so enzymes are specific, okay? They only bind to certain things. So they catalyze closely related reactions. They have what's called an induced fit model. Anybody know what that means, induced fit? So induced fit means that if I'm the enzyme, let me light this up a little bit. Okay, so here's the enzyme. Maybe that should look the same. Okay, and here's the molecule that it's going to bind. Induced fit says he's going to modify just a teeny tiny bit to exactly fit this guy, okay? So it's not as big of a change as what I've drawn, but he will modify to fit that. So it's an induced fit. All right, so it says the active site precisely fits the contours of the substrate like a lock and key, okay? So this guy has to modify a little bit so it fits so much like a lock and key. We know that we can't just go outside and stick our hoopty key in a BMW, okay? So it says keys match lock. Okay, so it says these enzymes are specific, so they'll catalyze only a few closely related reactions. So it says that's the reason why this induced fit kind of works, because it's going to be something really close to this that's going to fit this. So in other words, we know that if you are lactose intolerant, that means you don't have what enzyme? Lactase, okay? But if you have sucrase, can sucrase break down lactose? No, because we're saying these enzymes are specific for the reactions that they catalyze. So lactase splits lactose into two molecules, which are glucose and galactose. So we're saying these are specific types of reactions. So you'll see here's the molecule, and then it kind of, so you can see that he may have changed just a little bit to make sure he fit this molecule. The transition state, you can see that there's actually being stress placed on that bond. And remember, we're going to have enzymes that are going to come in here to help break that bond, removing or adding back. Remember, water is going to be split somewhere, and the enzyme is going to help to put the hydrogen back and the hydroxyl group back. And then down here, you'll see that the two former disaccharide has been made into two monosaccharides, and then you'll see that these guys are released, and then the active site goes back to its shape. Am I past my time? Okay, just checking. All right, have a good time at the dock. All right, so synthesis. Don't write this down. This will not be on your test, but the reason why I put it in here, because we were just talking about how enzymes are specific. So we know synthases are going to be what? Involved in what kind of reactions? Synthesis or anabolic reactions, isomerases, that tells you that we're going to do what? Make woo? Isomers, absolutely. Glucose isomerase is an enzyme that bacteria make, which makes sugars much sweeter, okay? It's where you get that high fructose corn syrup from, because they have enzymes that kind of reorganize it to make it a little bit sweeter than normal, so that's not good for you. polymerases. Okay, so that says we're going to do what? Make some who? Polymers. So in other words, putting all those nucleotides together. This is the guy that's going to help to do that. Okay, and then nucleases are going to break down the nucleotides. All right, so you get the picture. Okay, next. Okay. Factors that affect enzymes. It says many need cofactors in order to be active. When we looked at hemoglobin a while back, we said in order for hemoglobin to be active, it needed what? Iron, absolutely. So the cofactors can come in two forms. They may be coenzymes or apoenzymes, okay? And they are usually organic but non-protein. So they're inorganic ions or these organic but non-protein molecules. Because we said that coenzymes, where did they fit? Which category of molecules did they fit into? Anybody remember? Which category of molecules do the coenzymes fit into? No. Read that. So what? A what? No, he said proteins, which is wrong. It just says they are what? Organic, but who? Non-protein. No. Beautiful from the back of the room. He said, what, y'all? Did y'all hear him? He said nucleic acids. I said the coenzyme, no, they're not metals. They are nucleic acids. Now, the inorganic ions are going to be your metals. All right, so the apoenzymes, these refer to the metals, like your copper, your zinc, your iron, so on and so forth. The coenzymes, we know, what are their job? You got it right on the test, Daniel. What do the coenzymes do? Beautiful. So they regulate or they move hydrogen ions and electrons from one part of the reaction. So look at this. That's what's happening here. The coenzymes' job are to come in here and pick up these hydrogen ions that are being lost, and they're going to move them from here to there, and we're going to make water. Okay? So that's what coenzymes do. They move hydrogen ions, and they also transport electrons. So they're not going to physically pick it up and carry it. It's just going to help release it and put it into the right spot so that we can build up a concentration gradient. All right, so it says examples are NAD, which is nicotinamide adenine dinucleotide, FAD, which is flavin adenine dinucleotide, and NADP, which is nicotinamide adenine dinucleotide phosphate. Only thing you have to know for the test is what? NAD plus, FAD, and NADP plus. You don't have to know their names. But you need to... Oh, we're going to tell you, I'm going to tell you what they do. Right, but you're right. Know that these are coenzymes and know what they do. They carry hydrogen ions and electrons from one part of the reaction to the other. Should have learned that last time, okay? And then we're going to find out in a little bit later, I guess in this chapter, but where and which reactions these guys can be found. So these two are involved in cellular respiration. This last one is involved in photosynthesis. And again, we said their job is to do what? Move who? Two things. Hydrogen ions and electrons. All right, another thing that affects how an enzyme works, we need vitamins. Vitamins are required for the synthesis, or I guess this is specific for the coenzymes, because you can't make a coenzyme without having the vitamins present. So the vitamins are necessary for the construction of the coenzyme. We know that coenzymes are going to affect how a reaction is going to proceed because they need to be there in order to move about the hydrogen ions and the electrons. So we need vitamins. It's unusually hot in this room today. Oh, I'm getting all the heat from something up here. All right. And substrate concentration. If you don't have substrate, what's going to happen? Nothing. If you got too much substrate, then you're just going to be stalled because the enzyme can only do so many at a time, okay? But without substrate, we know it's going to slow down. With the right amount, we're going to just keep going. Too much, it's just going to keep going at the same rate that it was until it catches up. All right, temperature. We know that anytime we're outside the operating range, so this is nice because you're going to get this before you actually go to lab. But anytime you're outside of the operating range, it says if you're too low, so if we're way too cold, the enzyme is not going to work. But it says if you're way too hot, the enzyme is not going to work. But if we're right in that range where it actually operates, then the enzyme can do its job. So we're talking about factors that affect an enzyme's function. Okay? It says anytime we're outside of the normal operating range, it says we end up denaturing the enzyme. We end up denaturing the enzyme. No, denature means to rip up or to damage. Yes, as a result of being, right. All right, so let me ask you this. When did we say a protein is a protein functions? When it's in what structure? And this will be the last thing that we do. Tertiary. Because we said in tertiary structure it has a what? What do we say it has? Good, beautiful. Daniel said an active site. The active site is what binds what? The substrate. Y'all stop moving for a minute. I'm not done. All right, so it says if we are too hot, too cold, too acidic, I'm going to go ahead and put that one in here, too basic, it says that we can end up denaturing the protein. So we say protein denatures. So if I'm denatured, what does that mean? I'm not what anymore? Natural. Does that make sense? So it says this guy has done what? Good. So it's back in its linear sequence, but depending on how much damage we've done, he's even possibly broke up into little peptide fragments, okay? So we already know that this cannot do what that did, nor this definitely. It depends on how long we were outside the operating condition. This probably can go back there, but we already know, will this go back there? No, that's broken. We can't do anything with that. So we'll stop here. Hope y'all have a great weekend. Study, study, study. Yes, sir.